Molecular mechanisms of muscular contraction. Stimulus is received from the human. For example, noticing the ball is coming towards you and you thinking about wanting to kick it. This sends impulse from the sensory neuron from your eyes to your brain in your central nervous system. This goes from your brain to your spinal cord, all the way down to the nerve cell in your leg that you want to use to kick the ball. This goes into the motor neuron, which goes to the effector, which is to your leg. The motor neuron travels to the muscle. When it reaches the axon head, the nerve cell head, the impulse travels down the axon, travels through the myelin sheath, and this increases the speed of the transfer of the electrical impulse. These depolarizations at these nodes are known as the nodes of Rampeer. This is where the myelin sheath is not present. The process of saltatory conduction occurs in the myelinated nerves. The impulse jumps from node to node where depolarization takes place. As it's an insulator, it creates a faster transport of the impulse. This electrochemical impulse jumping from the myelin sheath through the nodes is known as saltatory conduction. The wave of depolarization as an electrochemical impulse is released. As the impulse reaches the edge of the nerve cell, it reaches the presynaptic membrane gap of the synapse. As it reaches the presynaptic terminal, this is the neuromuscular junction between the axon of the motor junction and the muscular fiber. This is a close-up of the section. So this is the presynaptic terminal and this is the presynaptic membrane. This presynaptic membrane, once it reaches here, the ACH vesicles transport down the synaptic vesicles containing ACH, which is acetylcholine, which is a neurotransmitter, transmit this neuron across the synapse to the receptor sites. This is the postsynaptic membrane to the receptor sites. Once enough ACH reaches across the synapse, the electrical impulse can travel across. This causes a contraction along both sides of the fiber. This is called the all-or-none contact. Hundreds of these neuromuscular junctions work together to create hundreds of muscular fiber contractions along the whole fiber. This happens so many times that the contraction can be strong and weak. The neuromuscular junction is where the motor neuron communicates with the skeletal muscle fiber. The axon terminal is the motor end plate of the neuron, with an enlarged end that contains the vesicles for the neurotransmitter. Acetylcholine is known as ACH, which is the neurotransmitter that crosses the synaptic cleft, which is across this area here. As the ACH is secreted into the synaptic cleft, it binds to the receptors on the motor end of the plate, the muscle fiber. If there is enough secreted, the electrical charge is high enough, the muscle action potential is created. Neurons stimulate the sarcolemma by generating an action potential, which is the electrical impulse down the muscle fiber. The cleft and the motor end plate contain acetylcholine, ACHE, which breaks down the ACH once the impulse has been passed on. This section is about sliding filament theory. This is the theory of the thicker sarcomere sections of the microfibrils intertwining, sliding into each other, creating contraction. So at the sarcolemma, these sections and the transverse tubules. The sarcolemma, the fine transparent tubular sheath which envelops the fibers of the skeletal muscles, storing muscle glycogen and myoglobin, has the openings across the surface that lead to the network of transverse tubules or T-tubules that allow for electrical stimuli to reach deep into the metal fiber. That is these orange sections here. These are the T-tubules. The sarcoplasm is the water solution containing ATP and pharyngens as well as the enzymes. That's this part here. Where is it? Sarcoplasm. Sarcoplasmic reticulum is under here. Sarcoplasm is under this part. The specialized smooth endoplasmic reticulum is expanded end that is next to the T-tubule, known as the terminal cisternae, with high concentrations of calcium ions. This is stimulated to release the calcium plus ions for muscle contraction to occur in the fibers. So now we go to the microfibrils into the muscle fibers. There are hundreds and thousands in each fiber. Each myofibril in each myofiber has thousands. These are circled by the T-tubules. Circled by the T-tubules go around in each fiber. There are bundles of very thick and thin myofilaments. The actin molecules are found in the thin filaments, these sections. The myosin molecules are found in the thick filaments, which are the middle parts. This part. And they are contractile proteins that shorten are responsible for contraction. So these are the sarcomeres, this whole section here where this slits in with the Z-line, M-line, and the other Z-line. This is the smallest functional unit of the skeletal muscle fiber, formed by the repeating myofilament arrangements. Each microfibril has about 10,000 sarcomeres that are thick and thin filament arrangements and are produced using the striated appearance of the fiber. This is the striated part, the Z-lines. This is what causes it to be striated. The overlapping filaments allow these lines and bands to slide in and out of each other to overlap so they can contract and shorten in during the contraction. Sarcomere lines. So the Z lines are the Z lines. These are the zigzag parts on each section. Z lines right here. These are the thinner filaments, both ends of the sarcomere, either end of the sarcomere. The other protein that connects the Z lines to the thick filament to maintain the alignment, the M line, is the middle. The M line is the middle. These that make the connections between the thick and the thin filaments. The sarcomere bands, on the other hand, contain the thick filaments. This is the A band. The A band is this part. The I band contains the thin filaments, which include the Z lines. The I band includes the Z lines. Now we get to the actin side. This is the actin filaments. These are the thin actin filaments. These are the twisted proteins that have the active sites for the myosin to bind to. The active sites are right here. These are the balls, the active sites. This is the strand of actin. When resting, the active sites are covered by the strands of tropomyosin. These are held by the troponin. In contrast to this, here is the thick myosin filament. This is the whole myosin molecule, thick filament. The head here, and this is the longer tail. The thick filament has the tail with the globular protein structure head that attaches to the active site during a contraction. These are much larger than actin sites. As the A band, thicker filaments go out, the myosin head contracts in, pulling in the Z line, gets released after ATP has been released onto it, comes back out, ATP is released onto it again, contracts down, pulls the Z line in, more ATP is released onto the myosin head, released again, and this happens till the full contraction. The sarcoplasmic reticulum stores the calcium ions. When contracting, the muscle fibers exert the pull. This shortens them in length. This is caused by the interactions between the thick and thin filaments in each sarcomere. This is the sliding filament theory. Calcium ions need to be present to trigger this. The contraction itself requires the presence of ATP. This is what energy is used for. So in short, as the action potential travels over the sarcolemma down into the T tubules, this triggers the calcium ions in the sarcoplasm to be released. When they are released from the sarcoplasmic reticulum, they diffuse into the zone of overlap and bind into the troponion. This changes its shape, altering the position of the tropomyosin strand, allowing the active sites and on the actin molecules to be exposed, so the myosin heads can bind to the active sites, creating a cross bridge. They rotate and cause the actin to slide over the myosin. ATP is required in this for energy. The sarcomere shortens. This repeats all along the microfibers, so the fiber shortens. ATP is required through each stage of this. Using this diagram, you can show the observations of the sliding filament theory. You can show the changes of the thick and thin filaments. The H band and the I band get smaller. The H band and the I band get smaller. The zone of overlap gets larger. The thinner filaments and the thicker filaments overlap more, so they move closer together and the overlap is larger. They overlap even more than they did before. The Z lines move closer together, with the width of the A band remains constant throughout the whole contraction. This is the relaxed sarcomere at rest, showing the locations of the A band, Z band lines with the I bands. During the contraction, the A band stays the same width, but the Z lines move closer together as long as the I band gets smaller in width. When the electrical impulse ends, SR recapture the calcium ions. The troponin and tropomyosin complex will cover over the active sites, recover them. So this has to happen again for a new contraction to even begin. This is when the contraction ends. As the sarcomeres contract, so does the entire muscle fiber. As the fibers contract, tension is created by the tendons pulling on the bones. Movement will occur only if the tension is greater than the resistance. The amount of cross bridges determines how high the level of tension is in the product. The variation in this is based on the amount of overlap of the myofilaments and the frequency of the stimulation. Therefore, if more calcium is built up, there will be a higher tension in the contraction. The contractions vary the tension based on the frequency of the fiber stimulation and the amount of fiber stimulated.